Then our next speaker is Dr. Qian. She's a postdoctoral fellow at the Center for Sleep and Cognition and Center for Translational MR Research at the National University of Singapore. So the floor is yours.
Good afternoon, everyone. My name is Xing Qian, and I am a research fellow from Helen Zhou's lab in National University of Singapore. Today, I'm going to present my work on brain structure-function decoupling underlies the cognitive development in youth. As we know, cognitive function undergoes extensive and projected development in youth prior to declining later in life. As one of the critical cognitive domains, executive function, which refers to a set of mental skills, including working memory, self-control, and cognitive flexibility, develops rapidly in preschool and continuously increases well into adolescence. Particularly, as an important process of executive function, cognitive flexibility supports almost every complex human behavior by enabling the switching between the modes of mental function to achieve goals. Deficits in its development are associated with lower academic achievement, risk-taking behaviors, and most major neuropsychiatric illnesses. Indeed, the age of onset for most of the neuropsychiatric disorders falls during childhood or adolescence. Studies have demonstrated that the projected development of brain structure and function within distributed association networks coincides with the improvements in executive function. Indeed, from this figure, which shows the important events during the development, we can see that the prefrontal cortex, which also refers to the executive control network, and the parietal and temporal association cortex, which also refers to the salience and default mode networks, undergoes projected changes during the development, providing an extended window for myelination and synaptic pruning. When looking at the time course of major developmental changes, including cortical thinning, myelination, white matter development, and functional refinement, we can find the heterogeneous changes between the lower order sensory motor regions, which are in yellow here, versus high order association regions, which are in purple here. Here, the darker shading indicates larger magnitude changes. These timelines review prolonged association cortex neurodevelopment marked by substantial changes in late childhood and adolescence. In recent years, the study of the brain anatomical constraints on its function has become increasingly important for our understanding of the brain-wide dynamics. The basic idea is to apply graph Fourier analysis to decompose the brain structural network into harmonic components that reveal global network organization. So here you can see the components are ordered from those with smooth spatial signals to those with varied spatial signals. Then we can decompose the functional signals in terms of these structurally informed components represented by a combination of these weighted components. This cartoon figure will give you a more intuitive explanation. So we can split the decomposed functional both signals into a proportion that aligned well or coupled with the underlying anatomical network, which means the signals in nodes that highly connect to one another exhibit similar signals to one another, and another proportion that diverged liberally or decoupled from the network, which means the signals in nodes that highly connect to one another do not exhibit similar signals to one another. Using a task-based fMRI, functional liberality from the anatomy was found to be related to the cognitive flexibility in healthy young adults. When introducing the structural decoupling index, or SDI for short, defined as the ratio between the norms of decoupled and coupled signals, it was revealed that the structural function decoupling is hierarchical across the cortex. Specifically, the spatial distribution of SDI aligns well with the functional hierarchy. As we can see, regions involved in the immediate perception are more strongly coupled, including primary sensory motor networks, while the regions involved in the complex cognition are more strongly decoupled, including the default mode, control, salience, and the limbic networks. Indeed, the correlation between the first functional gradient and the SDI is significant at the group level of the young adults. Using the HCP young adults, the accuracies of the cognition prediction using the SDI of the primary networks were lower than those of both the association network and the whole brain, which indicated that high-level association structure function decoupling contributes more than the lower-level primary decoupling to the individual cognition. However, only sparse data exist regarding how the white matter networks constrain brain dynamics in neural activity underlying the cognition during human brain development. And most of the developmental MRI studies are cross-sectional and predominantly focus on the middle childhood and later due to the challenges of acquiring high-quality data in younger children. To fill the first gap, our first study is to examine how the brain structure function decoupling changes across the development from children to young adults by leveraging on the HCP and NKI development datasets. Using a 419 region of interest postulation template, we extracted the bold time series from the functional fMRI data and constructed the structural connectivity matrix using the diffusion MRI data. By applying the eigenvector decomposition and the graph Fourier transform, we decomposed the bold signals into the coupled and the decoupled components, and we calculated the structural decoupling index as the ratio between the norms of decoupled and coupled signals across time. So higher values indicate that functional signals are more decoupled or liberal with the structural network, and the lower value indicates functional signals are more coupled or aligned with the underlying structural network. Then we calculated the global and the network level structure decoupling index by averaging the values across the whole brain or within each network. We examined the age effect on decoupling index at multi-scales using the generalized additive models. Using the HCP dataset, we found that the global SDI increased with age, suggesting greater decoupling at older ages. When navigating at the network level, we found the global increase was driven by the increasing SDIs of default mode, control, salience ventral attention, dorsal attention, limbic, and subcortical networks. In contrast, the visual network showed a trend of decreased decoupling with age. In addition, we consistently found a varied SDI across different regions with increased decoupling observed along the sensory association cortical hierarchy. When replicating using the NKI dataset, we consistently find a region-dependent age effect at the network level. Specifically, the default mode, control, and the subcortical networks showed increased SDI with age, while the somatomotor network showed a decreased SDI with age. And the SDI of the whole brain increased with age in the younger participants and trended to decrease with age in the older participants, which seems driven by the limbic network. Next, we want to know how the regional changes relate to the cortical hierarchy. So we modeled the age effect at each ROI level. We plot out the age splines and the ROI level age effect versus ROI transmodality. The age effect was calculated as the delta R squared between the full game and the nested model without the age term. The ROI transmodality was calculated as the mean value of the functional gradient template from the young adults within each RI. From the age splines and the age effect of the transmodal regions, including default mode, control, and the limbic networks, we observed increased SDI with age in these regions, suggesting greater decoupling at older ages. This increased decoupling might allow more flexible communications supporting complex cognition, subserving executive, social-emotional, and mentalizing functions. In contrast, the SDI of the most unimodal regions, including somatomotor and visual networks, decreased with age, which might allow more immediate perceptions. Next, as we have observed that the SDI was hierarchically distributed along the unimodal-transmodal hierarchy, so next we calculated the similarity between the individual SDI map and the FC gradient template of young adults and modeled how it changes with age. As a result, we can see this similarity was increased with age, indicating the spatial distribution of SDI develops towards the organization of the functional hierarchy during the development. When replicating using the NKI dataset, we have the similar findings. To summarize, we find increased structural function decoupling in transmodal association regions and a trend of decreased decoupling in unimodal sensory motor regions during the development. These changes lead to a pattern of structural function decoupling developing towards the organization of functional hierarchy. Together, these changes might allow more efficient perception and flexible complex cognition, supporting the cognitive development. As has mentioned earlier, there still remains an important gap in our knowledge regarding the typical development of brain in preschool children, which is a blossoming period of cognitive development, and how the longitudinal changes of brain supports the cognitive development remains largely unknown. To fill this gap, our second study aimed to examine the longitudinal changes of brain structure function decoupling in preschool children and how the changes support the development of cognitive flexibility. We studied children at 4.5 and 6 years old from Gustod cohort, which is the largest healthy development data set in Singapore. After careful image QC, we have in total 73 children who have good data at both visits. To measure the cognitive flexibility of the children, we conducted the dimensional change card sort task. Specifically, the task contains two paradigms, which are emotion and orientation. In each of the paradigms, the children need to match the cards by color or by emotion or orientation or by mixed rules. We think the performance of the mixed trials can reflect the cognitive flexibility the best. From the behavior results, we can see the task performance was improved at 6 years old compared to 4.5 years old, as the response time was reduced and the accuracy was increased for the mixed trials of both paradigms. This indicated that the cognitive flexibility was improved during the preschool children period. Using the same approach, we calculated the global and the network level structure decoupling index and examined the time effect on decoupling index using the linear mixed model. When examining the time effect of the structure decoupling index at the whole brain level, we found that the mean structure decoupling index was increased from 4.5 years old to 6 years old. The higher SDI value indicates that functional signals are more decoupled with the underlying structural network. Narrowing down to the network level, we found that this increased decoupling was driven by the higher order cognitive networks. The functional bold signals in salient ventral attention, executive control network, and the default mode network became more decoupled with the structural connectome from 4.5 to 6 years old. If you still remember the age splines of SDI modeling in development I showed earlier, you can see our findings in preschool children are consistent with these trajectories of SDI in the transmodal association regions. Next important question is how the changes in SDI support the improvement of the cognitive flexibility. So we examined the association between the changes of DCCS task response time between the 2 visits and the changes of SDI in each of the 3 networks showing time effect between the 2 visits. We found more increase of the SDI in executive control and the default mode network were associated with more reduction of emotion task response time, which means more improvement of the cognitive flexibility. This indicated that the executive control and default mode networks specifically played an important role in the development of cognitive flexibility, which is consistent with the previous findings in young adults that association region SDI contributes more to the individual cognition. In summary, the functional signals became more decoupled with the structural network during the preschool time, especially in higher order association cognitive networks. And this increased structural function decoupling supported a cognitive flexibility development in the preschool children. In conclusion, our findings revealed the differential trajectories of regional structural function decoupling in development, which strengthened the understanding of the brain structural constraints on its functional dynamics. Our findings also provide further insights into the mechanisms that take place in the structural function interaction relevant to the cognitive development. Looking ahead, we identify several directions beyond the current research. First, it will be interesting to examine how the SDI trajectory goes in middle and older ages and the role of structural function decoupling in cognitive decline. Secondly, we can investigate how the brain structure function decoupling in disease cohorts deviate from the normal trajectory. And thirdly, we can explore the genetic and molecular contributions to brain structure function decoupling and how the environmental factors interact with these contributions. To close my presentation, I would like to thank my mentor, Prof. Helen Zhou, and all the lab members. Also, I'd like to thank our collaborators from NUHS and SICS. At the end of our lab also do some work on the functional organization in the children and the psychosis. Although the poster session is ended, welcome to chat to my colleagues in the next few days. Yeah, thank you.
All right, thank you very much. We do have some interesting questions in the app. So the first one, is there any specific toolbox or software that directly yields decoupling strength? Is there a toolbox available to do the calculations?
Yes. So the original paper is published in the Nature Communication 2019. They have published the code of the structural function decoupling, so you can download from the GitHub. Yeah.
And thank you. The second question is, how do you hypothesize structure function decoupling progresses into old age? Is a reduction in executive function driven by decoupling?
In the age cohort, right?
So into old age, I think you have covered childhood, but how would you suggest how would you hypothesize that it would evolve in old age and older people?
In the aging, right?
Yep.
Oh, so my hypothesis is that the changes of the structural function decoupling in aging is region dependent. So maybe it's the opposite trend as the development. Maybe in the higher order cognitive networks, the decoupling will be going down as the age goes going up, and in the unimodal regions, maybe the perception, the decoupling will going up. Yeah, that's my hypothesis.
Thank you very much.